good afternoon, or I might even say good evening. I'd like to thank Doug and Bonni Goldstein and the organizers of the conference for inviting me to moderate. I'd also like to thank the remarkable panel that has agreed to share their knowledge with us today. I'm a retired cardiologist who first became interested in cannabinoid oncology when I saw Mara Gordon show brain scans from patients with brain tumors who were treated with cannabis and had well-documented shrinkage of the tumors. When I saw those images, I knew that's what I wanted to do. Mara was kind enough to teach me her method for approaching patients with tumors. I saw many patients. Many patients were able to shrink their tumors, and many were able to achieve no evidence of disease. However, I saw far too many patients who failed to respond to cannabinoid antineoplastic therapy. About a year ago, Dr. Meiri gave a talk to the Society of Cannabis Clinicians, and his presentation stimulated me to reassess the therapeutic cannabinoid protocols that I had been using. I'm gonna mention your names now and just sort of wave your hand to introduce yourself when I say your name.
I don't know what you correct, so I'm afraid.
When I was asked to moderate this panel and select the panelists, I thought that we would learn the most by merging the intellectual capital of the best researchers, Dr. Meiri, Dr. Sanchez, Dr. Russo, and clinicians, Drs. Hergenrather and Goldstein. An aspect of Dr. Meiri's work also dealt with the use of various solvents, and so I wanted Dustin Mahan, the CEO and Chief Science Officer of Endocanna Labs in Colorado, to show us extraction results of several chemovars with multiple solvents. Laboratory work that we have available in this country, we don't have mass spectrographs as Dr. Meiri has at his disposal. Dr. Goldstein and Dr. Hergenrather will share their experience first, hopefully speak to their failures as well as their successes in using cannabinoids in the treatment of cancer. Then Mr. Mahon will show us some comparisons of various cultivars and multiple solvents. With that background, we will then begin asking the full panel the questions detailed in the panel description. I hope you have those at your disposal now. Otherwise, I'll be reading them as we go. And the objective of the panel is to learn how we can better apply preclinical research to improve patient outcomes. So with that, if you would start, please, Dr. Goldstein.
Good evening, everybody. So I'm going to talk about my experience with mostly pediatric patients that I treat with cannabinoids for cancer. Remember, cancer is a term that we use that encompasses a whole bunch of different diseases, and as Dr. Sanchez and Dr. Meiri's research shows, that we can have breast cancer, we can have colon cancer, but there's still subtypes and they may not respond the same way. So it's a very difficult, complex question when you're approaching patients that come into your office and are asking for help. A few of the concerns with pediatrics is that their same chemotherapies are being recycled over the years, very few new treatments for these kids. Also, unfortunately, pediatric cancer only receives about 4% of all cancer research money. So there is 96% of the cancer research money goes to adult cancers, and so that's also kind of hindering research. I will tell you that also what I'm finding is that when parents come in with their children, one of the biggest concerns is my child may survive, but what is the quality of my child's life going to be down the future? Is my child going to get a secondary malignancy from radiation? Is my child going to have cognitive impairment from chemotherapy and radiation? Is my child going to have cardiac disease? And I was preparing for this panel. I actually read about a gentleman who's doing research at University Hospital in San Antonio, who he himself is in his 40s, and he is a cancer survivor. And he is in his 40s, has had severe repercussions from the chemotherapy. He's had a cardiac arrest, he's had strokes, and so he is actually studying what chemo does to those who survive from childhood cancers, and he says it accelerates the aging process. And if you've seen some research in the last couple years about cannabis, we know that it can help actually kind of reverse aging as well. So when I have a patient that comes in with cancer, of course I go through a history physical exam. I talk about with the family what their goals are with cannabis therapy. For some of them it is just symptomatic treatment, because they're on a protocol that may give them very good survival. Some of them want to add cannabis as a adjunct antineoplastic agent. And so I often discuss both treatments with the families. Many of them do opt, even if they start with symptomatic treatment, many of them, as they see that their child's responding really well, then they come back and say, you know what, we want to up the dose, we want to start giving our child more cannabis to see if we can try to increase the chances that our child's going to survive this. With children, I use all the cannabinoids. They're obviously the research that Dr. Meiri and Dr. Sanchez are doing is very helpful, but still to this day we don't know what combinations are going to kill which cancers. So as we're going to discuss, I think other panelists are going to probably say the same thing. I have found that combination treatment is clearly the way to go. I usually use 5 different cannabinoids, ones if you heard me speak yesterday, so CBD, THC, THCA, CBDA, and CBG. I try to use as many different preparations and also from different companies, because then I can be sure that they're probably going to be grown a little bit differently. They're going to hopefully cover a lot of the different compounds. Right now, I kind of look at this as, you know, somebody comes into a hospital who's really sick and you do not know let's say you know they have an infection, but you don't know what they have. You may send a culture and you cover them with what we call broad spectrum antibiotics till you can narrow down what's going to work. And that's kind of how I approach this, broad spectrum, using multiple different compounds to try to see what will work. And I start low dose and we titrate up, of course, always concerned about side effects if there's going to be any. I will tell you, children, because I have taken care of adults with cancer too, children tolerate cannabinoids way better than adults. They don't drive a car, they don't go to work, they don't have to make dinner, so they kind of go with the flow. Many of them, I will tell you, the parents will tell me there is absolutely no side effect and their child goes from being sick on chemo or side effects of radiation. I had one patient that went down from about 90 pounds down to 57 pounds post-radiation, and she's gained it all back just from adding cannabis in. So children really tolerate cannabis well, and in my experience, I am seeing just over the last few years that more and more patients or parents are bringing their children to me earlier in treatment rather than waiting until they've been told that there's no other treatment. Many of them want to help their child get, of course, the best results. So that's all I have to say. I don't know if there's anything that you want to add, Joe.
Well, you'll have a chance to weigh in later if you want.
Okay. Just one other thing. Joe did ask me to talk about possibly the patients that don't respond. So sometimes people get to my office so late, like unfortunately I had a child show up on a Wednesday and the child died Friday. That's obviously not enough time to be able to intervene, although the family didn't even have time to get oil. I've had other patients come in who Clearly, it's towards the end, and I don't think that it's going to reverse anything, but certainly quality of life at the end of life so that the child's out of pain, maybe eating a little bit, maybe more playful, certainly makes a difference for these families. There are a few cancers that I have not got good results with. DIPG, if you've heard of that, it's a very rare brain tumor. Unfortunately, we have not gotten very good success. Patients who have leukemia seem to respond really well. So, of course, it also depends on the kind of cancer in terms of response and also at what stage they are when they get to me.
Thank you.
Okay. Well, I wanted to first thank the organization for the opportunity to participate. It's wonderful to get together with our colleagues from around the world and meet new folks in the field. I've been a cannabis doc for a long time. I started my career about 40 years ago on a commune in Tennessee learning about cannabis as medicine, but cancer wasn't part of that at that time. It wasn't until I opened my office about 20 years ago up in Sebastopol, California, that I began to see cancer patients on a regular basis. At that point, there were somewhere in the range of 8 to 10% of my patient population came with cancer. And since that time, really in the last 3 years, this demographic has changed drastically to where now there are about 30 to 40% of the patients that come in to see me are coming because of cancer, and mostly asking for treatment that may mitigate, may help in slowing the progression of these tumors. That's a huge difference, and I think those of you are going to see the same kind of demographic in your practices. I have seen some amazing things over the years, and some of the first ones were smoking a small amount of cannabis on a daily basis to see a glioblastoma never recur. For 8 years, or for 9 years, I might add, before that point that the patient stopped smoking cannabis and he had a full recurrence of the tumor within a year. I've also seen a neuroblastoma disappear after UCSF had given up on him, and they sent him home and said, comfort care, we don't have anything more to offer. And then he too, as a 9-year-old, began to smoke a little bit of cannabis for pain relief, and within a couple years, the tumor had disappeared. Malignant melanoma is another case where I've seen them disappear. These tumors disappear in multiple patients over a period of time. And since these other tumors, hepatic carcinoma, other people with glioblastoma, and really multiple tumors have done well. But saying that, I also have to say the majority of patients with cancer don't see this kind of response. Of course, I'm not controlling for either the cannabis or for the treatment protocol. I might advise a treatment protocol, but I have no idea what they're going to do once they go out into the back to home and try to find these products. So approaching a patient with cancer, I think, is very important to just be, as you know, the ethical approach is to say, look, I expect that we're going to get treatment benefits in terms of symptomatic cures or symptomatic relief. You're going to have a better appetite. You're going to have some pain control. You're going to sleep better. Your mood's going to be better. The anxiety is going to go down. Nausea and vomiting are going to be controlled. We can expect that in most all patients who use cannabis at all. But whether or not a tumor is going to regress, that's a whole other thing. And, you know, it's looking at the work of Dr. Sanchez and Dr. Meiri and others before who have really led us to say, well, you know, it was McAllister's work 7 years ago, 8 years ago, who said, hey, THC and CBD work together better. There's a synergy of these molecules. So this has led us along the way to directing patients to some at least hope that we're going to find a combination of cannabinoids that may work best. So at the least, I want symptomatic relief. At the most, we may get lucky and be able to treat a cancer really effectively and slow the progression or in a rare case, see a tumor disappear. For those patients that come to me and they're already chronic cannabis smokers, I say, look, you know, it doesn't really look good because this tumor has developed in the face of a regular use of cannabis, and we can certainly try high-dose therapy, and we often do that. So that's getting up into these gram quantities that have kind of gotten known as the Simpson oil, and that protocol of taking a large amount of cannabinoids. I can't attest to any results for that, but people are at least trying it. We also are still faced with the issue of people come in and say, what is the best strain for me to treat this cancer? And we simply do not have that information yet. We're close, as we can feel the research coming. I know people are going to be looking for THC and CBD and CBG, and from this point on, they may also be looking for compound 33118B.
Right.
You know that I didn't say the right one, right?
Oh. Okay, I'll scratch that out of my notes. So method of administration, I think, is very interesting because we do know that a puff on cannabis has a very high blood level for a short period of time. And it was the great work of Donald Tashkin, sitting in the front row, who taught us that, yes, this does provide some injuries, some irritation to the lung and the airways, but we're not going to be causing COPD and emphysema and significant alterations in the pulmonary function studies. So, I don't discourage people from smoked or vaporized cannabis as a method of administration. Otherwise, if we're going to be treating with cannabis, we tend to look to the oral forms in order to get the big doses that we think might be necessary. Again, some of these cases that did so well earlier, 15 years ago in my career as a cannabis specialist, were very small amounts in many cases. And so I don't know if we're way overacting to be giving gram quantities of cannabinoids. We may be, but there seems to be information that we don't really fully understand yet, and that would be markers of the tumors that would give us some clues as to what tumors are going to respond best to what cannabinoid profiles. Dosing and frequency, this is tailored to meet the patient's needs. It really has to do with what kind of tumor we're dealing with. If it's a slow-growing tumor and they just want to come in and have an idea as to what to do, it may be a small dose of cannabinoids at bedtime that's sufficient. If we want to ramp it up, we might give a big dose of CBD in the daytime, even twice in the day, the morning and the afternoon, and have a more sustained blood level. So now you can get up into hundreds of milligram quantities of CBD-rich full plant extract cannabis oils. If they can tolerate the THC in their product, then fine. I think it's probably a good idea until we learn better. But for a bedtime dose, I'm really encouraging people to use a combination of THC and the other cannabinoids. Acid cannabinoids, I think, are certainly of value too, and I do talk about the green cannabis as well. If they have friends that are growing, put those buds in a blender and have them on a daily basis if they possibly can. The duration of action is different because of these P450 cytochromes that are a little different from patient to patient. Some people are going to probably be adequate with a dose a day, and others may need 3 doses a day in order to optimize or maximize their cannabinoid therapy. So I'm excited to be in the field. I do the best that I can, and I'm really relying on these colleagues of mine in this field to guide us as to what might be best in the future. And it's one thing to grow them in a lab and kill cells in a laboratory on a petri dish, and quite another to be able to do it in vivo in people that are dealing with these cancers. So that's more or less the way I'm approaching cannabis therapy of cancers, and I'll welcome the opportunity to answer questions if they come along. Thank you.
Thank you.
Hi, my name's Dustin Mahan. I'm the president, CEO, and CSO of EC Labs in Denver, Colorado. The last 2 years, I was out in the crowd with you guys watching these great minds up here. I mean, I'm so humbled right now to be up here with this panel that words can't say enough of how thankful I am to Dr. Joe and to all the people at CanMed here. Cannabis has saved my life, and it has fascinated me ever since I jumped into to understanding it. I did a quick experiment here to try to help the panelists and understand a little bit more.
There you go. You got to go back now.
Okay.
Which way am I? I don't know which way I'm going.
Sorry.
There you go.
Okay. So we are extraction experts at in Colorado. We use hydrocarbon extraction and some ethanol extraction. I did use a lab white labeled to do some CO2 extractions for me here. So there's still much more research that needs to be done from the cannabis plant on what things do what here, but we wanted to focus specifically on the cannabinoids and the terpenoids. Cannabinoids and terpenes have shown largely to have significant impact on cancer cells. There are far too many variables right now to validate really a best classification for solvents. However, this experiment will provide information on the best solvent for specific applications like total cannabinoid potencies or efficiencies. Right now, we have an inability to test all of the great things that my friend here gets to test with cannabinoids and terpenes, but it leaves us with probably more questions than we have answers, but this is as far as we've gotten, and we really need a lot more research. So the few things that we look for out of the cannabinoid extraction or from extraction of the cannabinoid plant is going to be efficiencies. So how much of the cannabinoids and the terpenes did we pull out of the plant material, as well as potencies. So how pure is the concentrate that we get out of it. The other thing that I like to look at is the decarboxylation rates. You know, the plant grows in the raw acidic cannabinoids, and certain extraction techniques actually will activate those cannabinoids. So the different variables that I tried to accomplish here were going to be using a quick wash versus a soak, which is going to be plant material on a quick wash is contacted in very minimal exposure time, trying to increase the purities, but you're going to sacrifice some efficiencies, versus soaking the plant material where you're contacting with the solvent for prolonged periods of time, trying to pull as much of the cannabinoids and terpenes out of the plant material, but understanding that you'll probably pull some impurities as well. So we took two different chemovars of cannabis. We took the high THC strain and a high CBD strain. Here we ran them through several different methodologies of quick wash and soak with 5 different solvents. And we had the cannabinoids and terpenes potencies tested on the starting plant material, the concentrate, as well as the ending plant material. So we used propane and butane. We used ethanol, and then we did a subcritical and a supercritical CO2 extraction. One of the major questions that I get is how much solvent is left over. Every state that's legalized has set some sort of a residual solvent level. Even the United States Pharmacopoeia set a residual solvent level that they could have in pharmaceuticals. However, I'm a huge fan of believing that you should really not have any residual solvents left over in your product. And I have proven, and it is possible, to remove the solvents from all the different methodologies that I've shown here. So it's a quick test that we get in Colorado from RM3. A lot of NDs up there. I could flood your inbox with NDs, but I think we can all agree that it's very possible to get all your solvent out of there. So the different cultivars I used, the first one, this is my THC strain. This is our alien rock candy, somewhere in about 6% terpene content, 22% cannabinoid content. That's your cannabinoid test from the alien rock candy. And then my Spectrum strain. Spectrum's a very high CBD strain. We grow it out to a little over the 0.3% THC mark, but it's used heavily in Colorado as a hemp strain. So RM3 did some testing for us, and the things that we were able to figure out, well, we wanted to look at what solvent extracts total cannabinoids with the highest potency here. So on the THC heavy strain, we found that supercritical CO2 actually tested at the highest purity at 85.33%. Propane quick wash, which is minimal soak time, which makes sense. We hit 83.81%, and the butane quick wash at 83.59%. On the CBD strain, your propane and your supercritical flipped, but they're pretty negligible in potencies there at 81.8 and 81.76, and then the propane soak came in 81.01. So the way I looked at for highest efficiency isn't necessarily how many grams you pulled out of there, but how many milligrams that you pulled out of there. So multiplying your grams by your potencies there. And on your heavy THC strain, we found that the butane quick wash came in at the highest efficiency, and ethanol soak right behind that, which again within 1%, so pretty negligible. And the propane quick wash came in right after that. On the CBD strain, we had a really, really great extraction with the ethanol soak. Supercritical CO2 came in after that, and then a butane quick wash. As far as terpenes go, propane quick wash won out on both sides there. The ethanol, we had a soak for THC and a quick wash for the CBD, and then the ethanol quick wash again for THC and a butane quick wash for the CBD. And then as far as the extraction efficiencies, propane again looking really good for your terpenes coming in, ethanol and butane right behind that. So the only thing that I recognized in the testing for decarboxylation rates was that it was the CO2 process that tended to decarboxylize the plant material. Now I understand there is specific ways that you can run your machines. We ran these on a very commercial, very standard, supercritical, subcritical, and we saw that supercritical across the board decarboxylized both THC and CBD about 60%. The interesting thing, though, on the subcritical run is that your THC in your spectrum, THC jumped about 20% and the CBD jumped 40%. However, the THC in the CBD strain jumped 40%, but the CBD in that spectrum strain only jumped 2%. So I theorize that it's actually the different pressures that are extracting in that subcritical, the difference between subcritical and supercritical being the pressures, that our subcritical hit high enough just to activate the THC, but not enough to activate the CBD. So is there really a best solvent? I think what we have shown is that each solvent has a unique ability to do something different than its counterpart. We have shown that there are some better in others in specific categories, and we've also shown that there are far more variables in extraction that are still to be found out. I think that the fact that every body and every cancer is unique, that a combination approach is absolutely necessary and far more research is needed. Thank you.
Thank you. Can we turn that? That's one of your slides, right?
Yeah.
Right. I should have told you beforehand. But anyway, can we turn the screen off for now? So with that background, what I'd like to do is ask some questions of the panel. Dr. Hergenrather alluded to the ratio of THC to CBD. How do we know when to give more THC? Are there situations where we want to give more CBD than THC? How do we learn how do we decide how much of each cannabinoid to use? Anybody?
In a situation like that, if I were asked that question by a patient, I would try to go to the literature and see what Cristina or Dedi or Manuel Guzman has done previously that indicated that there was a preference for a particular cannabinoid, or whether there actually had been combinations tried before. Additionally, I have the benefit of having a large network of friends that I can ask about this kind of thing. I would additionally look for any synergies that might be present in the terpenoid literature. But as we've seen, sometimes it's not so clear. But first thing is to see what has been done or what even might have been known historically. And what history tells us that a lot of treatments of tumors in the past were with what appeared to be raw cannabis, whether it would have been CBDA predominant or THCA dominant. And as we found out in the last year from another group in Spain, Nadal et al., that THCA is a PPAR gamma agonist. So that's a nuclear receptor that affects gene transcription and may have a very prominent role in treatment of cancer, but that still needs to be investigated further.
Thank you. I might add that one of the techniques that I have been attracted to is that we know from Dr. Sean McAllister's work that CBD downregulates the ID1 gene, and the ID1 gene is associated with tumor growth and metastasis. So if searching the literature shows that a particular tumor routinely expresses a high level of ID1, that might be a good indication for a good response to more CBD.
Your mic's breaking up.
Pardon me?
Your mic's breaking up.
I know. I know. So, how often should the THC be taken? In my experience, it's easy to give the majority of the THC at bedtime because the patient can sleep through the psychoactivity. But Dr. Goldstein referred me to an article of a patient with lymphoblastic leukemia who expressed blast cells that were not curtailed when the THC was given twice daily, but when it was given every 8 hours, the blast cells fell. So I would like to ask the question of the panel, how often should we give the THC? And when?
You want me to, I can answer. I can tell you what I do. So for most of my patients, again, with children, it's a little bit easier because they're usually not losing function that most of us would not want to give up, i.e. driving a car, functioning. If the family expresses to me that they want their child to still go to school and to function well at school, I will have them take THC in the afternoon when they get home from school and then in the evenings. If it's a child who's not going to school, which, as you know, if they have cancer, many of them are not attending school, I usually recommend THC every 8 hours along with the other oils, CBD and the raw cannabinoids, and now I've added in CBG as well. I'm probably, and I tell people this, I'm aggressive and I often overtreat, but I'm terrified to undertreat, to be perfectly honest, because, again, children tolerate cannabinoids very well. And I find that really the only downside is the cost. In most families, if we're getting somewhere with the cannabinoid treatment, if we actually can get to a point where the cancer is gone, we can back up and go to more affordable doses. So again, much of it, if they're very small, it doesn't cost that much to give a fair high dose. But in general, in children, again, THC, I often recommend that every 8 hours. We don't have a lot of clinical, really very few clinical trial experience to really hang our hat on how long the cannabinoids are going to hang around in any particular person. With Jeff's experience with patients just smoking and getting results, certainly we know that when you inhale, it doesn't last in your body very long, and certainly not at high doses. So it certainly has to do a lot with the sensitivity of the tumors as well to exactly what they're taking.
I would concur with Dr. Goldstein. If I can get a patient to take a THC, CBD, balanced medication in 3 times a day, I'll encourage them to do that. If it doesn't work into their life plans, if they're going to pick up the kids at 2 in the afternoon, then take the CBD in the morning and reserve the THC for later in the day. So I do encourage that. Often, right from the start, in California, under the laws that we have today, I tell people right from the get-go, you need to start thinking about growing your own medicine. The cost of these molecules have gotten so expensive. They come in thinking they're going to be taking a dropper full of cannabinoids 3 times a day, and I say, no, we're talking about an ounce bottle of cannabinoids a couple times a day. cost is absolutely prohibitive in this kind of dosing. So I really encourage people to talk to friends and consider growing their own medicine. Again, multiple cultivars I think are very important, and the opportunity again to get acid cannabinoids into the mix I think also is really valuable.
Thank you.
So I want to say something, maybe to be the bad boy on this panel. We don't know. That's the truth, okay? So the answers here was how to give cannabis that the patient will be comfortable. We're not treating patient to be comfortable. We want to kill the cancer. So when a physician gives chemotherapy, he's not answering if you can drive or not drive. We don't know. Every answer will be good as the other answer. We don't know what the patient needs to get. We don't know if it's accumulating the tumors. So in the mice, I can tell you that they see accumulation of cannabinoid in the tumors. It's like stuck there. Is it this true for patients? Do we need THC in the evening? Do we need CBD in the morning? The truth is we don't know. So every answer will be as good as the other answer. Cristina, Sanchez, and me, like the scientists, we know that we cure every type of cancer in mice. So it's not true for people. So there is a huge gap between killing cells in the plate, killing tumors in the mice, and treating a person's. And the correlation is so far that even if we learn about it in the mice, we won't know how to treat the patient before we'll do real clinical trials checking this kind of thing. So the truth, in all these conferences, people are coming and asking what we need to give to the patient. First of all, we don't know really that it's working. We are assuming, we're hoping, and even if we know, we don't know what to give them. That's the truth. That we can't run around it.
Thank you. How did there? No. So I have another slide. I have the slide from your most recent paper in Nature, and what you showed, which is impressive and so important, and I'm so thankful that we have this. That you compared 36 cultivars versus 94 cannabinoids, and you can see from this top part of the chart that there are different cannabinoids in different cultivars, and they're all over the place. And also, I stole this slide from your Patients Out of Time presentation in May, and this shows that this shows 6 different solvents, and this confirms what Dustin found. So the solvents here, the first 6 down here are the same as the 6 across the top. And here are 2 cultivars extracted with 6 different solvents. And what we can see is, for example, if we're looking for CBGV, we're not going to get it in this cultivar with any solvent. But if we take a cultivar that has that cannabinoid, we can extract it with only butane and carbon dioxide, not with ethanol, which many of the fine extracts are made with. So for me, it's pointing in the direction of making our medicine with multiple cultivars extracted with multiple solvents in order to get all of the cannabinoids. Now, Dr. Meiri showed us in his presentation this afternoon that there were a few key compounds that were necessary to kill the cancer. So I'm wondering if you would comment on the benefits or potential benefits or lack of benefit of making these combinations of multiple cultivars and multiple solvents.
Yeah, I'd like to take that one initially. For me, it's easy. There are only two possibilities, ethanol or CO2. I have little confidence in the ability to totally purge these other solvents, many of which are toxic. Additionally, there are a lot of people out in public that think that they can do a home extraction with butane and they blow themselves up in the process, as the story I told on Monday. The safe ones are ethanol and CO2 extraction. Obviously, for the latter, you need some very technical equipment, but I just don't see the place for the others myself.
Ethanol is just as flammable, and I've seen very big fires from ethanol. Just throw that out there.
Yeah, I don't recommend that patients do this at all, but...
I agree.
And if I can...
Go ahead.
If I can add something, I completely agree with Ethan. And we have to keep in mind that we are talking about patients that take this medicine on a daily basis, several times a day, during very prolonged periods of time. So if you have a tiny little bit of something that is below detection limits in your sophisticated equipment, but if you accumulate that little bit of something 3 times a day, every day, for months, for years, that tiny little bit becomes something important. So I completely agree with you.
So I just want to say it for my defense. First of all, it's like hitting the expert. I don't remember these figures. It's a long time ago. But second, this is academic work, and this is not coming to treat any patients. And for sure that there are solvents there which are poison, just to showing that different solvent create different extracts. It doesn't, I don't want nobody to treat with these solvents patients. I think what Christine and like the people here said, it's extremely important. When you're doing an extract, you need to think about the solvent, what is left with the patient. And regarding your questions, so when you are mixing different cultivars, you're diluting compounds. So it's true that you have higher variability, but you're diluting the compounds. And again, I don't have the answer what is good and bad to the patient. I just know what is the action that we're doing and what is the result. And I think the physicians here have much, much more experience how to treat the patient. So Bonni and we were far away from knowing, you know, in this, we're just showing the complexity and how much we don't know, but we don't have good answers, I guess.
So Dr. Russo, would you think that combining CO2 and alcohol extracts might provide better patient outcomes?
Possibly, but again, you know, I'd want to see some evidence, a rationale behind combining to get those specific components.
So I want to say something about fire and ethanol. When I started to do extraction, I looked to find the method in PubMed. It's usually where scientists look for evidence. And there was no good way to do the ethanol extraction. So I went to YouTube's. And first of all, I thought that I'm the first one that doing ethanol extraction without tattoo. Second, the guy said the most important thing when you're doing the extraction, don't smoke.
Thank you. I wonder, since you used ether in your lab, did anybody go to sleep?
I use ether?
Yes. The first solvent, yeah.
It may explain why the student was very slow doing that.
One of the things that intrigued me from Dr. Sanchez's recent paper was the lack of efficacy in adding back the terpenes. Now, she added back 5 terpenes. 2 of those in Dr. Russo's publication on antineoplastic terpenes, only 2 of the 5 were designated as antineoplastic, linalool and alpha-humulene. So what that triggered in my mind is when we talk about the entourage, I've always thought of the entourage being the terpenes, but I'm wondering if it's not as much or more so the minor cannabinoids that make up the full entourage. And what I was thinking about in the combining the extractions from multiple cultivars and with multiple solvents was enhancing the entourage. If you had compounds that were not present in one cultivar, you could bring them into play, and even though it's diluted, if you standardize for THC and CBD, it's diluted, but maybe in that slide that the last presentation, the small amount of CBGA was what made the difference, and maybe it doesn't take very much. Maybe it's a catalyst rather than a true activator of. Anyway, I'd appreciate your comments on that.
For me?
You, yes, please.
Sure, it's an intriguing question. One of the things I noted when I read Cristina's paper from her group was that there was no detected limonene. Limonene actually has had observed activity, at least in vitro, in breast cancer. There was a study done in humans of high-dose limonene in breast cancer. Unfortunately, it didn't read out, and my impression of that was, well, they forgot the cannabinoids. Yeah, certainly when I thought it did relate to minor cannabinoids, and again, given what Cristina said in relation to my question, I have a strong hunch at this point that THCA is responsible for the difference from THC alone.
So I want to comment about the terpenes. I think we all think the terpenes are important. I'm speaking by myself. So I think the terpenes are important, but I know Ethan, what he's thinking, and I'm quite disappointing. So one of the first things that we're doing in the lab when we see a phenomenon, we cut the terpenes out to see if it's affected. And except for sleep disorders, that we have a mice model for sleep disorders that we do see the terpenes important, in epilepsy, in the cancer models, in the other models, I didn't find that they are important. So every time that we cut them out, it doesn't affect the phenomena, which is extremely disappointing for me. But these are the results for now. So again, except sleep disorder, I didn't find anything that they were in the mice model and the cell model that their terpenes was very affecting. So that's the results.
So a quick question. Is hemp a legitimate source of CBD in antineoplastic protocols?
I didn't, again, the question?
But
I didn't get the question. What?
I want to know if we can use hemp. It's very difficult for some patients in Europe to get high CBD cultivars for their medicine. It's virtually impossible. I do some consults from Europe or with patients in Europe. It's also difficult in many areas in the United States to get ACDC and other high CBD cultivars, and I'm wondering, but hemp seems to be much more available, and I've looked at some of the lab work on the lab analyses of hemp extracts, and there are some that do have a rich terpene profile and have substantial amounts of CBD. And I'm wondering if that is a potential source of CBD.
Maybe. However, I think they're going to spend a fortune in the bargain, given the kinds of prices I've seen. You know, a lot of this is a byproduct of prohibition. Let's be clear. You know, there are better ways. A dedicated, what we'd call drug chemovar, that's rich in CBD is likely going to be preferable. Plus, with a hemp extract, so-called, generally we're not talking about any significant amount of THC, and for most tumors that's a necessary component.
Right, right. I'm just thinking about the CBD component. Yeah.
What I've seen from a lot of CBD extracts is that in a raw CBD extract, an ethanol extract, if you will, is that you can see THC get into the 3 to 5% range on that, which for some patients, I've seen some really good responses to that one with some of my cancers, but like you say, it's still kind of out there. I would like to have a lot more THC, but for those people that don't have access to anything better, I can't see the harm, and I think it could actually help.
And so a follow-up question on that is, we've heard a lot of talk about CBG and CBGA at this conference, and it looks like it is turning out to be a useful cannabinoid that we ought to include in our antineoplastic protocols. And would hemp be a good source of CBG and CBGA?
We have one strain in the lab that have 17% CBG and without THC and CBD almost at all, like 0.2 CBD, 0.4 CBG, THC. And this is a very potent strain. So you see that there are cancers that are being affected by that. There is, in the epilepsy model, it's affecting, like there is many models affected. And again, I don't know if it's just the CBG, but for sure, this strain is very, very potent strain for many things, and I believe that CBG is as potent as THC in CBD. It's usually not in high percentage, but I guess, again, my belief that THC in CBD is a little bit covering for the others, but if you eliminate them, there will be other cannabinoids with high affinity to the receptors, and they can be very potent if they will be in the percentage of the THC in CBD as we usually see them. So it might be that there are other, what we call minor cannabinoids, that if they will be in high percentage, they have higher affinity to the receptors. So we call it affinity and avidity, how much it's binding the receptors. And we see that there is minor cannabinoids with very strong activity on the receptors, and just amount. I believe that in the cultivars and in the thing that people are doing, we will find more and more strains that have different cannabinoids that are expressed. There is already like THCV strain with 10 and 11% of THCV, which is extremely psychoactive. People think that just THC is psychoactive. THCV is also very psychoactive, but we will see more and more cultivars with different cannabinoid expression and different effects. That's, I already see that. People think that they can send everything to my lab in FedEx, and they're right. So I see a lot of things.
You want me to make a comment?
Go ahead.
Because of my work with children with autism and families, of course, searching for products that might help their children, I've learned about a couple of CBG, actually 3 CBG products available here in California. 2 of them are very, very low THC, so can be sold across state lines as kind of under the hemp. But we've gotten them tested at multiple labs, and one runs about 50% CBG with about 2% CBD and I can't remember what the THC is, it was very, very low. And one of the other ones I'm waiting on lab tests, and then one is a sublingual tablet that you can get in 3 milligram amounts, and we've have that pending right now in a lab test. So CBG is becoming available. I'm sure there's going to be more products on the market if you're interested in using it. And there is some an article, and maybe Joe, you can share that with people. The article that talked about CBG for colon cancer. Yeah.
So I'd like to go back to something that Jeff said about this. I blew this one out too, did I? Can you hear me? Yeah, you can hear me now. So you said that you suggested that patients throw a couple of buds in their smoothie in the morning to get the raw cannabinoids. There is a pretty extensive literature for the raw cannabinoids. The CBGA I think I found 5 articles showing that it had benefit for breast cancer. Dr. Meiri has shown some experience with THCA and prostate cancer, but in your slides, when you showed that in one of your YouTube videos, it turned out that THCA also killed the glioblastoma cells in vitro. Next to the decarboxylated THC, the THCA was the second most potent cannabinoid. So there's also literature on CBGA, and there's papers on colon cancer with THCA and CBGA, and so I'm wondering if we should include raw cannabinoids in a formal way in our cancer protocols. So I'm just envisioning that since they're not psychoactive, the patient could take the CBD and the raw cannabinoids during the day and take a big dose of THC at bedtime and get a really full entourage of compounds to fight the cancer. And I would like to hear the panel's response to that.
Why not?
Yeah, great. Share it around.
So throwing a few buds into the blender is a pretty good way to do it if you can't make the oil, and there's not many products available in the dispensary that have a full complement of raw cannabinoids.
So I want to say 2 things. Yes, there are cancers that have been affected by the acid form of the cannabis, and I don't know if it's the acid form or other terpenes. I didn't dissect it yet to know. The major cell lines that we checked were more affected by the decarboxylated, but there are a few of them, and prostate cancer is one of them, that being highly affected by the acid form. We need to remember that the acid form is the native form on the plant. So the plant not creating THC, it's creating THCA, and there is a logic to think that there is a role for that, in animal receptors, if it's kind of defense or attract mechanism for the plant. But usually they are less potent than the active form, like the decarboxylated form. That's at least
3 minutes.
Our results. If the patient can eat cannabis like that, it's not affected. So as Eitan said, why not? So I want to say, like, you saying things that are more related to United States. In Israel, we don't have a problem of the amounts of the patient. They are not paying for that. It's not related to a price. They are paying fixed price. Doesn't matter how much they are getting. They can get 600 grams a month or 2 grams a month. It's the same amount for them. We don't have a problem of CBD cultivars. They can choose. So every grower will have at least one high CBD cultivars, and they can choose and can have it. So all of these problems we don't have. It just we don't know what is good to give them. But if we know it was available for the patients.
So do you routinely use the THCA now with, or recommend it to patients with prostate cancer?
My patient have 4 legs and a tail, so they understand. I'm not a physician. I'm routinely try checking it in my lab on the models that I have. I'm not offering patient, and I'm trying to avoid it. I think, you know, people that are experienced and physicians should take these decisions.
We have 1 minute.
Can I make a quick comment?
Sure, sure.
So I think ultimately, you know, there's a huge amount of research that has to be done, including clinical trials. And one of the biggest problems, of course, is the Schedule I status. So any chance you get to try to talk to any politicians, if you know people, we have to get it descheduled so that we can do the research, period. We live in chaos where CBD is still a Schedule I drug. If it comes from a pharmaceutical company just recently, now it's a Schedule 5, but and then there's a patent right on THC by the government for neuroprotection, but yet it's still a Schedule 1. So it's utter chaos, and that has to get sorted out. But ultimately, having the science that Dedi and Cristina are doing is so very important, but at the same time, what we're being warned about over and over is not to take their results and then apply it to human beings, because that's not linked. It's the initial findings. What we're seeing is, sure, maybe it definitely helps in breast cancer cells in a test tube and then also in mice. But again, remember, we're missing clinical trials. For Jeff and I that treat human beings that come into our offices, we offer the best that we can. There is no one protocol that's going to work for everybody. I had the unfortunate, well, I guess it's a mixed experience of seeing 2 teenagers with the same exact stage 4 osteosarcoma. It's very similar stories. One young man, the family was very motivated. They took high-dose oils, and that young man survived, and he was diagnosed at age 14, was probably not going to make it till his 17th birthday, and he just turned 20 about a month ago. But at the same time, I had another case referred from the same oncologist, who the family came, and I gave a very similar protocol, and they just had a really hard time giving the oil, whether it was psychosocial situation or the child didn't want to take it, and that boy died. So you can't conclude very much other than there is certainly a chance, and what we're seeing, I know Dr. Hergenrather and I, better quality of life, maybe extension of life for certain patients, and certainly an ability to give somebody some hope and to move forward despite the diagnosis of cancer, and sometimes very devastating cancer diagnosis. But at the same time, there is no one magical protocol that seems to work for everybody, despite the fact that you may have the same cancer or the same type of cancer. You have to remember that we're still very early in the stages of this, and I don't think that, I'm sure a lot of people came to this panel saying, well, I want to know what to take or what to recommend to my mother-in-law, but at the same time, what I'll tell you is that there's no reason not to try if you certainly have an advanced cancer, or certainly if it doesn't look good for you.
Guys, we are out of time, but if you want to take one question from the audience, we can do that.
I want just to say that there is few clinical trials in Israel that's running now with the cannabis and cancer. There is like 3 Rick Simpson-like. There is one on glioblastoma, and I think this data that will come from there will start to give better hints. And I totally agree with Bonni that, you know, you can conclude for the mice and from the cell lines how to treat patients. But I think from this research it will be better. And I admire these physicians that treat the patient and take the advantage. As they said, it's not toxic, and there is no, it's, even if it's give a little hope to the patient, it's worth it.
Thank you. Thank you all for, in your own way, pointing out what we don't know, which I think forces us to zoom out and use more of a systems theory approach when it comes to working with patients. What I didn't hear, and I'm wondering if some of you could comment on, would be cautions or contraindications for using cannabis in patients that, say, might be doing immunotherapy or based on some animal data that just came out this month from Lopez Valero et al., showing that CBD may reduce the efficacy of temozolomide in glioma model in mice. If any of you have any cautions or contraindications to offer.
I can make a comment. So when a patient comes in, of course, they're required to supply me with the list of medications that you're taking, including any chemotherapy or any new immunotherapies. One of the things that I find helpful is that you can call a pharmaceutical company. They're always open to questions, and if you ask them, have there been any, of course, drug-drug interactions with anything, like CBD is usually the big one that we find interactions. And for some of my patients, if they're on a medication and their oncologist wants them to take that, sometimes I'll tell them, you know, you can take low doses of cannabinoids to try to mitigate side effects. However, it may not be in your best interest to take high doses of cannabinoids right now if it's either going to clear that drug out of their system quicker or actually allow that drug to accumulate and create toxicity. So we have to be very careful, because the vast majority of patients that are coming in now, and I'm sure Jeff can say this too, are choosing treatment. They're not coming at the last resort like it used to be maybe about 5 or 8 years ago. Most people that are coming in are actually coming in fairly early, and so now it's created this review of all medications, because many of them are interested in combination therapy. And that's why it's important that, especially now that there's legalization in our state here in California, that someone over 21 doesn't self-medicate while they're on a oncology protocol, because they could actually make themselves toxic on medication.
Let's have a round of applause for our panel.